So I want to start by saying something that 10 years ago we couldn't say, which is that we now have treatments that can work for patients with advanced metastatic cancer that are curative. Specifically, cancer immunotherapy can cure metastatic cancer. And I'm showing some clinical trial data here for those who may not be as familiar, and this is from patients with advanced metastatic melanoma, a form of skin cancer, the type of cancer that I treat in the clinic. And I'd just like to draw your attention to something for historical purposes, which is just a little over 10 years ago, a diagnosis of stage 4 or metastatic melanoma was essentially a terminal diagnosis, with less than 5% of patients being alive 5 years after that diagnosis. As of 2011, we had our first immune checkpoint inhibitor. These are monoclonal antibodies targeting co-inhibitory receptors on the surface of T cells. These invigorate anti-tumor immune responses, and this worked in about a quarter of patients. As of 2014, 2015, we saw the approval of checkpoint inhibitors targeting the PD-1 pathway, which demonstrated about a 40-45% long-term benefit in patients with advanced melanoma. And the combination of these two treatments results in over half of patients being alive greater than 6.5 years, now greater than 7.5 years after a diagnosis of advanced metastatic melanoma. And this really is transformative, just thinking how far we've come in just over 10 years in this field. So based on the success specifically of the immune checkpoint inhibitors targeting the PD-1 and PD-L1 pathway in cancer, we've seen a huge explosion of approvals of these drugs, not just in melanoma, but in lung cancer, kidney cancer, and a variety of other cancer types, to the point now that almost half of all solid tumor patients are eligible for PD-1 or PD-L1 based cancer immunotherapy. However, the proportion of patients that are actually benefiting long-term, similar to the previous slide that I showed you in melanoma, is still a fraction. It's a minority of all of these patients. At the same time, the market for these drugs continues to increase. If you look where we are now in 2023, with the global sales exceeding $40 billion estimated, in 2028 expected to exceed $60 billion. But the landscape of clinical trials in this particular space is extraordinarily complicated. And if one looks at this particular infographic, you can see in the bullseye the FDA-approved treatments. It's a select number. And then moving outward, the concentric circles demark the phase 3, phase 2, and phase 1 clinical trials, with the dominant space occupied by phase 1 clinical trials. And if you focus just in melanoma, it's pretty easy to appreciate that from the oncologist perspective, there's great challenges in trying to determine what's the most appropriate phase 1 clinical trial for a given patient without patient-specific factors to make that decision. And there are now over 1,000 clinical trials across different disease indications evaluating these different cancer immunotherapy targets, with many of these combination approaches having an immunotherapeutic drug partner. Unfortunately, to date, the combination efforts have not been that successful, and efforts to improve upon the current best immune checkpoint blockade in cancer immunotherapy have not worked. And shown here are the results for 2 phase 3 clinical trials that initially showed promise in phase 1 testing, phase 2 testing, and ultimately were shown to be no better than the standard of care, a single-agent anti-PD-1 antibody, pembrolizumab. But most of the new trials continue to be combination trials. And you can see back to 2014, 2015, around the time of the approval of the first PD-1-based immune checkpoint antibodies, the number of combination trials has continued to increase year by year, dominating the vast majority of the clinical trials still. And the need for novel combinations isn't just important for patients who need more effective treatments, but it's important for drug makers as well, and even the drug makers for some of these FDA-approved indications, as these drugs eventually will come off patent, and many of them are seeking combinations to extend sales. So one thing that has been agreed upon in the field is that we need better models. We need improved models of human tumor immunity. And this is the #1 challenge facing the field of immuno-oncology, facing clinicians and especially cancer researchers. But we need preclinical models that translate to human tumor immunity. And one can envision that having such a model would help in drug development and discovery to de-risk emerging targets and understand and examine heterogeneity of therapeutic responses, to deprioritize ineffective strategies earlier in their development, and helping to refocus the portfolio on more promising approaches and targets, and ultimately to identify biomarkers of response and resistance to these novel treatments, ultimately in the hopes of reducing the failure rate of early phase clinical trials. And some drug companies, for example, Roche has already started to bet big on personalized approaches to try to use patient-derived model systems as part of their efforts for drug discovery and development. So our solution are PDOTs, or patient-derived organotypic tumor spheroids. Organotypic is a term that means two or more cell types grown in a three-dimensional space to mimic normal human physiology. And this is a novel biomimetic technology platform to study the dynamics of tumor-immune interactions in a patient-specific manner, essentially offering a window into the tumor-immune microenvironment where we take a biopsy from a patient, disaggregate it, dissociate it, and embed it in a microfluidic culture device so it can grow in three-dimensional space similar to a normal tumor. And shown here is what we actually see. And you can see that we preserve the immune microenvironment in our PDOTs, where we have the tumor cells in green, the immune cells in purple, and specifically PD-1 on a subset of those immune cells. And we've already started to bring PDOTs to bear on some of our preclinical efforts evaluating combinations. And I'll walk you through this slide from a recent publication where we performed a phase -1 clinical trial of an established drug targeting anti-PD-1, and in this particular waterfall plot, each bar is an individual PDOT specimen from a different patient. If it's going up, it's not working. If it's going down, it's working. And the different colors indicate different tumor types. In teal is a mismatch repair deficient colon cancer, one of the tumor types with the greatest response rate to PD-1. In yellow is kidney cancer, and black is melanoma. We can see that our novel target, which was a hit from a screen, this inhibitor targeting tank binding kinase 1, showed improved single agent activity in this cohort of 30 PDOTs, but the combination, this novel combination, was more effective than either single agent alone. And specifically, we were able to demonstrate activity in clinical specimens from patients who had already seen these immune checkpoint inhibitor antibodies. And this is from a former patient of mine that was clinically resistant to anti-PD-1 and anti-CTLA-4 immune checkpoint inhibitors in the clinic, but was sensitive to the TBK1 inhibitor and specifically the combination. And the same story was true for a handful of other specimens, just showing you two here to represent that. And we see there's great potential for functional precision cancer medicine using PDOTs for evaluating novel immuno-oncology targets, small molecules, antibodies, antibody-drug conjugates, and even cell therapies like chimeric antigen receptor T cells, but specifically for testing rational combinations. And that can be done across cancer types and across cancer stages far faster than can be accomplished in phase 1 clinical trials. But there's also a longer-term value proposition, being able to use PDOTs to predict in a patient-specific manner which patients are likely to benefit or not. And these are two representative PDOTs from patients with melanoma that benefited and were sensitive to immune checkpoint blockade, ICB, one that was resistant, and the PDOT's responses mirrored the in vivo clinical efficacy. And our goal is to establish academic industry alliances to support drug development, preclinical, and clinical testing. And we think insights from PDOTs will guide preclinical and clinical development with ultimately the goal of reducing the failure rate for phase 1 clinical trials. So for immediate opportunities and next steps, we've already founded a company as of 2019, and that's Xfira Biosciences, that's based here in the Seaport. And Xfira is already offering ex vivo profiling to multiple industry partners and clients in the goal of predicting patient responses to oncology therapeutics. This was co-founded by myself and my postdoctoral mentor, David Barbie, and a colleague, Cloud Powlitz. There is IP protection that is held and currently licensed by Xfira. Series A fundraising is ongoing. And this is also an ongoing partnership with AIM Biotech, a company founded by Roger Kam at MIT that is the manufacturer of the microfluidic devices that we use for our PDOTS cultures. In addition to that, we think there's potential value in developing an academic industry alliance, specifically finding the right partnerships so we can leverage the unique strengths of an academic cancer center like Mass General, and specifically an academic cancer center like the Mass General Cancer Center, to leverage the insights from our lab and the technology to develop these alliances to facilitate and accelerate the identification of novel and effective immuno-oncology strategies to ultimately provide that long-term clinical benefit that we see in a subset of our melanoma patients, not just to more melanoma patients, but for all cancer patients. With that, I'll end. Thank you.